Thank you. So MMPs are, okay, apparently my slide does not want to change. There we go. MMPs are matrix metalloproteinases, play important roles in the wound healing and tissue remodeling process. In fact, they degrade multiple extracellular matrix proteins as well as inflammatory mediators. So the quality of the scar after a myocardial infarction or an MI depends on this equilibrium between ECM degradation and ECM synthesis. If you tip the balance towards degradation, this can lead to cardiac rupture, whereas if you tip the balance in the opposite direction towards synthesis, this can lead to wall stiffening and therefore decreased cardiac function. So MMP9 is one of the key regulators during the post-MI remodeling process. During the first week, MMP9 doubles in the plasma beginning at day 1 post-MI. There's multiple MMP9 substrates that have been found in vitro. This includes collagen, fibronectin, elastin, as well as inflammatory mediators such as CXL4, interleukin-1 beta, and as well as interleukin-8. So our team uses proteomics approaches to detect changes in ECM after a myocardial infarction. We have had to overcome a couple of different issues, mainly due to the relatively low abundance of ECM proteins, as well as the poor solubility. We have focused on different attempts. We have focused our attempts on methods in order to reduce the sample complexity. So in the past, we've used multiple approaches to do this. We first tried to use natural enrichment by focusing only on the infarct region at the later time points post-MI. We then started testing different decelerization buffers and found that using 1% SDS was the best. And we then used separated into a soluble and an insoluble fraction to help after decelerization to help with enriching for ECM. All of this then culminated into what we call our Texas 3-step protocol, which uses a non-denaturing buffer to remove the soluble fraction, followed by a decelerization to remove the cellular fraction, and then the insoluble proteins were then underwent acid extraction, deglycosylation, and solubilization before we ran it through the mass spec. So while the Texas 3-step did improve our ability to identify ECM proteins within a sample, because of the large number of steps, this made it less ideal for quantitative comparisons. So more recently, we have started using glycoproteomics. We did this started with a collaboration with Johns Hopkins Center and the Weizong Lab. We used glycosylation or glycoproteomics because ECM and membrane proteins are frequently glycosylated. So this was a way for us to enrich for our ECM proteins. So on the figure on the left, this is shows the collagen changes post-MI. So after an MI, the scar is mainly composed of ECM and collagen proteins. On the right, this is an example of how we use glycoproteomics to enrich for extracellular matrix proteins. This was actually in patient plasma from a sepsis study that we published back in 2015. So in using this approach, we were able to enrich where 74% of the proteins identified were in the extracellular region. So our hypothesis was that we can use glycoproteomics to be selective for ECM, that we can then this will allow us to better analyze the infarct post-MI. So we used C57 wild-type mice and MMP9 null mice. We used 16 mice per group. The mice underwent coronary artery ligation and were sacrificed at day 7 post-MI. Our outputs were we looked at echocardiography, we did glycoproteomic profiling using the insoluble fraction of our infarct tissue, and then we used some bioinformatic techniques to analyze the proteomics data, as well as immunoblotting and in vitro cleavage assay to validate the proteomic findings. We then used immunohistochemistry, immunofluorescence, phagocytosis assay to identify functional rows of these proteins that were identified by the mass spec. So echocardiography showed that post-MI you have a decrease in fractional shortening, an increase in your diastolic and your systolic dimensions, as well as a decrease in your infarct wall thickness. When we deleted MMP9 in our MMP9 null mice, this was attenuated. So this is consistent with what we have observed, as well as what others have published in the past. One thing to note is that infarct size was consistent between the both groups. So this is not due to the initial ischemic injury, but was actually due to the remodeling process after the MI. So for our glycoproteomics, we took both wild type MMP9 null for male and for female. These were the day 7 MI infarct that we then went and fractionated into an insoluble fraction. The glycoproteomics, we used the solid phase extraction and linked glycopeptides method. The samples then underwent LC-MS-MS, and peptides were identified using Proteome Discover, using the sequence database, and quantification was then performed using Steve, and then we then confirmed using immunoblotting in vitro assays. So here is just a little summary of our glycopeptide data set. As you can see, 78% of the peptides that were identified belonged either to the plasma membrane, the cell surface, or the extracellular region. We then looked at whether these were actually some of the peptides that were identified were actually glycosylated. So the LB sample was an incubated with or without PMGAS-F, an enzyme that releases the N-linked oligosaccharides, and immunoblotted for both fibronectin, gamma-sarcoglycan, or periostin. And then as shown here, you can see there was a shift down in the molecular weight, which confirms that these were all glycoproteins. We then used bioinformatics analysis and clustered the samples. And as you can see here, what is shown in the green is our wild type. What is shown in the orange is our null, and they cluster fairly well with the exception of one wild type and one null, suggesting that there was definitely a specific pattern when we were deleted MMP9 null. However, when looking at the clustering by sex, as you can see, male was shown here blue, female is shown here in pink. There was no clustering, suggesting that there was no sex-related differences identified by this analysis. So we found 45 peptides that had both twofold change and were significantly different between wild type and MMP9 null. 36 of these 45 proteins were found to be within the extracellular region. Of the 45 proteins, cartilage intermediate layer proteins, CILP, and platelet glycoprotein 4, or CD36, were found to have the highest fold increase in MMP9 null compared to the wild type animals. Because we know that post-MI proteins are not static and they change over time, we then measured the time course of both CILP and CD36 over the time course post-MI. And interestingly, CILP actually increased over time and was higher in MMP9 null compared to the wild type. So this was not what we would expect to see if, for example, CILP was actually an MMP9 substrate. So we can say that this most likely CILP is not a substrate of MMP9. When we look at CD36, we found that over time in our wild type animals, there is a significant decrease in CD36 post-MI, where in the MMP9 null animals, these levels stay pretty comparable to what we saw in day 0. This is more of what we would observe if we thought CD36 was an MMP9 substrate. So we then looked at the 45 different proteins by string PPI software using a high confidence score, and we visualized that by Cytoscape. So many of the changes that were actually observed were found to interact with each other, as shown here by the interconnecting lines. The strongest associations are shown by the thicker lines. The red nodes are the ones that were upregulated in the wild type compared to the MMP9 null. I'm sorry, that's opposite. Upregulated in the MMP9 null compared to the wild type. The blue are those that were downregulated. The gray nodes were not identified, but were connected to the 45 proteins that were identified. And then MMP9 is shown here in green, and our two proteins of interest, CD36 and CILP, are shown here in yellow. So one thing to note is that MMP9 actually already has a known connection to CD36 through thrombosbondin 1, where CILP is actually not really shown to be interconnected with the proteins that were identified. Because CILP was shown to actually increase post-MI and most likely was not MP9 substrate, and because it has no known connection to MP9, we then therefore did not evaluate this protein any further in the study. So we showed that CD36 decreased over time post-MI. We then wanted to confirm that CD36 was actually an MP9 substrate. So we performed an in vitro cleavage assay where we incubated recombinant CD36 with active MMP9 for 24 hours and then visualized this with immunoblotting. And as you can see here, when both CD36 and MP9 was shown in combination, you have a bunch of CD36 fragments that show up. In addition, we then want to confirm if this was also true in vivo. So we looked at CD36 at the lower fragment, at the lower kilodalton area, and found that there seemed to be a 35 kilodalton fragment that showed up in our wild type, but did not show up in our MMP9 null. So confirming that CD36 is both an in vitro and an in vivo substrate of MMP9. So just a little history about CD36. It is also called the fatty acid translocase. The N-terminal fragment of thrombospondin 2 has actually been shown to bind to CD36. This stimulates apoptosis. In addition, CD36 is expressed by both neutrophils and macrophages. So because we had this increase in CD36 in the nulls, we wanted to see if there was also a difference in apoptosis, seeing as CD36 is known to regulate apoptosis. So we performed immunoblotting for cleaved caspase 3 in the infarct tissue in our wild-type MP9 null mice, and found that cleaved caspase 3 was increased in both groups. However, it was elevated in our MP9 null mice. At day 7, the neutrophils are becoming apoptotic and are being actively removed by macrophages. So we then wanted to see if this increase in apoptosis is actually maybe due to neutrophils. And we found by neutrophil immunohistochemistry that neutrophils were lower in our MMP9 null group compared to our wild type group, suggesting that neutrophils are actually being cleared out of the infarct area at a higher rate with MMP9 deletion. To then confirm that the increase in caspase 3 was due to an increase in apoptotic neutrophils, I performed dual immunofluorescence for neutrophils and tunnel. So tunnel is a marker of apoptosis, and the overlay analysis shows there's a higher number of apoptotic neutrophils in the MMP9 null group compared to the wild type group. So this demonstrated a new signaling role for MMP9 in the regulation of apoptosis. We then looked at isolated out blood neutrophils and stimulated with either MMP9 or used a CD36 inhibitor to look at the apoptotic process in the blood neutrophils. And compared to unstimulated cells, MMP9 decreased neutrophil expression of caspase 9 and increased caspase 3 expression, but had no effect on the other two apoptotic markers. Where when we use the CD36 blocking peptide, this increased expression of caspase 3, in addition to the apoptotic inhibitor XIAP, indicating that while MP9 does seem to regulate apoptosis, it doesn't seem to be doing this through CD36 degradation. So in addition to neutrophils, as I mentioned earlier, CD36 is also known to be expressed by macrophages. And in fact, CD36 has been shown to be required for phagocytosis of apoptotic cells. So we then wanted to look at CD36 expression on the macrophages to determine if it's actually higher on that of our MP9 null mice. So by immunofluorescence, we found that using MAC3, which is a marker for macrophages, there was no actual difference in the number of macrophages in wild type compared to MMP9 null. However, again, there seemed to be a higher expression of CD36 on the macrophages of our MMP9 null mice compared to that of our wild type mice. So to test then if MP9 then could regulate macrophage phagocytosis, we then isolated out macrophages from the infarcts of our MMP9 null and our wild-type mice. The macrophages were then stimulated with or without MMP9 and incubated with fluorescent labeled particles for 2 hours. And then we quantified using a spectrometer and visualized using immunofluorescence. The right panel just shows a positive control. So in this sample, we actually used IL-4 stimulated macrophages and exposed to the fluorescence beads. And as you can see, the macrophages that engulf the beads show the green fluorescence. So macrophages isolated from the day 7 MMP9 null infarcts actually had an increased phagocytic potential compared to those of the wild type animals. When we then exposed these macrophages to active MMP9, it reduced phagocytic potential in both wild type and MMP9 null mice or macrophages. This data is in line with our previous experiments, which indicated that MMP9 cleavage of CD36 is actually decreasing macrophages' ability to remove apoptotic neutrophils. Thus, MP9 deletion is actually accelerating the resolution of inflammation. So just to summarize this study, we showed that with an MI, you have this elevation of MP9, which then leads to increased proteolytic cleavage of CD36. This diminishes the neutrophil apoptosis as well as the ability of a macrophage to phagocytose and remove neutrophils, which then associates with our increased LV dilation. When MP9 is deleted, CD36 is preserved and leads to increase in phagocytosis to coordinate the removal of neutrophils. This then serves to resolve inflammation at an earlier and attenuate LV dilation post-myocardial infarction. So in conclusion, using glycoproteomics, we were able to enrich our extracellular matrix, which allowed us to then use functional proteomics to reveal a new signaling role for MMP9 as far as its regulation of inflammation. Thank you.